Alrighty, hello everyone. As we continue on our discussion on lipids and biological membranes, we're going to take a look specifically at membrane proteins. And there are three different types that we'll consider: integral membrane proteins, lipid-linked proteins, and peripheral proteins. And what we'll notice that each of these three different types really differ in the nature by which they associate with the membrane. Integral membrane proteins tend to be tightly associated with the membrane via hydrophobic interactions. In lipid-linked proteins, just like the name may imply, they're linked to the membrane via or anchored to the membrane via a lipid, right? And the complexity level varies. And then we have peripheral proteins that are loosely associated with the membrane, mainly via electrostatic interactions and some hydrogen bonding, right? And when we take a look at those proteins as we proceed later in the course, we'll notice that they have numerous functions, whether it's catalyzing chemical reactions or mediating the flow of different molecules across the bilayer and acting as signaling molecules as well. One example, or the first example, is integral membrane proteins, also known as intrinsic proteins, which are embedded within the lipid bilayer or within the membrane, which we know is hydrophobic in nature. So they could be removed from the membrane by reagents that overcome the hydrophobic effects, right? So right here shown is a figure as a surface diagram showing you the surface of the protein, where you have neutral atoms shown in like white or gray, and then you have positively charged residues shown in blue. Those would be lysine or arginine that are positively charged under physiological conditions. And then you have the negatively charged, which is shown in red. You can think about it as aspartic acid, so aspartate, glutamate, right? And then it's showing you the lipid molecules to resemble a lipid bilayer, but it's showing you the green being the hydrocarbon and the red being the oxygens, right? And then some phosphorus that is, don't know if you could actually see it, but it's right here in orange color. So integral membrane proteins are said to be either what we call monotopic, meaning they'll only interact with one side of the bilayer, or they could be polytopic, that span the entire membrane. So one example that we could draw of an integral membrane protein, again, they're also known as intrinsic proteins. They could be associated with the lipid bilayer only on one end of the lipid bilayer, and then the rest of the protein is exposed to the outside. So that's an example of monotopic, right? So only a single segment. It could be on one end of the bilayer, or it could be on the other end of the bilayer, but it's still referred to as monotopic. So those, you could think about them as being mono, so only one section of the protein is embedded within the bilayer. Monotopic. Or it could be known as polytopic, and in polytopic proteins, the protein spans the entire lipid bilayer, right? So one part of the protein is exposed to one side, the other part to the other side, but it spans the entire lipid bilayer. And those are known again as polytopic. And everything is through hydrophobic interactions. So if you think about it, the lining of the protein that is facing the lipid bilayer would tend to be hydrophobic amino acid residues. So we can identify the location of those different domains or regions within the protein with respect to the lipid bilayer, right? Is it monotopic? Is it polytopic? Which side of the protein is actually embedded within the lipid bilayer? We can figure those things out by what we call membrane protein topology, right? So we use topology, what we call a topology specific reagent, a reagent that will interact with only the side of the protein that is exposed to the extracellular side of the membrane, or one side of the bilayer, right? So we use a reagent that reacts with the protein but do not cross the bilayer. And then we could be able to identify the different domains or regions with, that face either side of the bilayer. And one example is using proteins is reacting the protein with an enzyme such as trypsin that will cleave the protein, but it will not penetrate the lipid bilayer. So it will only cleave segments of that protein that are exposed to one side of the bilayer. And then it would allow us to identify which domains or regions within the protein are exposed to the outer or extracellular face of the membrane. And generally we call them topology specific reagents because they allow us to determine the different regions or domains within the protein with respect to the bilayer. And one example or classic example that we'll take a look at that use topology specific reagent is erythrocyte glycoprotein. It's a transmembrane protein that spans the lipid bilayer, meaning you have one segment of the protein on the extracellular side or outside the cell, one segment of the protein is within the lipid bilayer, and one segment of the protein is inside the cell. That's a single polypeptide. If you take a look specifically at glycophorin, we have its N-terminal domain, right? So that's the N-terminus right here, but we're going to call this its N-terminal domain. Its N-terminal domain is on the outer surface, right? And then it was cleaved by trypsin, right? So that's how we know this segment of the protein was on the outer side of the cell. And it's also shown linked to a number of different carbohydrates, right? So those carbohydrates, if you notice, are linked to different serine and threonine residues. And you have one right here that is shown linked to an asparagine residue. So you have both N-linked and O-linked oligosaccharide. In any case, it's C-terminus and is on the inside. So that's the C-terminus. So we're going to call this entire thing its C-terminus end is on the inside of the cell and cannot react with that reagent. So it cannot react with trypsin. So it cannot be cleaved by trypsin. And then if you also notice the N and the C-terminal domains, they both domains contain polar or charged group or hydrophilic groups, right? And they also contain some hydrophobic groups as well. But then if you go in and take a look at the segment of the protein that is embedded within the lipid bilayer, then what you'll notice is that particular segment is mainly hydrophobic, right? You have phenylalanine, valine, methionine, alanine, glycine, valine, isoleucine, right? So it's primarily hydrophobic in nature. It's around residue 75 to 93, roughly speaking, right? It starts this particular segment, right? It's about 75, and those numbers are just indicating the residue number. So the glycophorin is 31 amino acids long. All right, so if you also look at the orientation of the glycophorin in the membrane, it's really asymmetric, right? Part of it is, it's in different parts within the membrane, right? But its amino terminal segment is always on the outside, and we're going to learn about that on our next video. And if you think about it, the glycosylated domains are always going to be on the extracellular face of the bilayer, right? Which could have a number of different functions. So some polytopic protein, which we said span the membrane, have a single hydrophobic sequence in or around the middle of the protein, and that was the example we just saw. You have a single hydrophobic segment that is, around the center of that particular protein. So glycophorin was one example, but it could also be either the C-terminus or the N-terminus end of the proteins. And some protein might have multiple hydrophobic segments that span the lipid bilayer. They might span it once or several times, and typically those have, in terms of secondary structure, either alpha helices or beta sheets. So those regions, so how can we identify those regions within the protein? So they're identified or were determined experimentally by measuring the free energy change, or delta G, that might be associated with transferring amino acids from water or from a non-polar solvent into water. So the movement of an amino acid side chain from a hydrophobic solvent into water could cause a reaction or a measurable delta G that is either highly exergonic, right, when we have polar charged residues, or highly endergonic when we have non-polar residues. And this is shown right here. If you have polar charged residue, it's highly favorable because you're moving them into water and the molecules are polar. Whereas if you have molecules that are highly non-polar, that would be a highly endergonic reactions. So there is a cutoff, which is about 85 kilojoules per mole, right? So a region, and then you compare that to the actual sequence of the protein. So if you have a given sequence of a polypeptide chain, you take a look at the number of amino acids within that polypeptide chain, and what you're looking at is the free energy of those change associated with those particular amino acids. And then if you have a region that has 20 or more residues of highly hydrophobic residues, and we can relate that into something that has a hydropathy index higher than 85 joules, and then it's a region that's at least 20 amino acids residues long, then this region of the protein or segment of the protein is presumed to be a transmembrane segment. And overall, hydropathy index is quite accurate, right? So those are really indicating the amino acid residue, the residue number within the polypeptide. Residue 50, residue 70, 10, 11, whatever. If we have a segment within the protein that's at least 20 amino acids long and has, a hydropathy index, higher than 85 kilojoules per mole, that is predicted to be a transmembrane segment. So it's a method that allows us to identify which regions within a protein is likely to be embedded within the lipid bilayer. All right, so those transmembrane proteins could be part of integral membrane proteins if they span the entire lipid bilayer, and they have to satisfy their overall energetics in terms of hydrogen bonding within the polypeptide instead of within water molecule, because now they're embedded within a hydrophobic region, right? They tend to be difficult to crystallize because they're overall hydrophobic. And one of the best studied polytropic tropic protein is really bacteriorhodopsin. It's a single polypeptide that folds into 7 alpha helical segments, which are shown right here in different color, each of which really spans the lipid bilayer, and they're clustered perpendicular to the lipid bilayer. So if these are like the hydrophilic or the head groups of the lipid bilayer, right? I'm just pretending to draw a lipid bilayer, and then you have the fatty acid chains. So this is the lipid bilayer, right? So I'm just going to go ahead and draw it in yellow. So the protein those alpha helical segments are clustered perpendicular to the lipid bilayer, right? Or to the plane of the membrane, which is a common motif found in membrane proteins involved in signal reception. All right. Bacteriorhodopsin is a light-driven proton pump. It's densely packed in Halobacterium seleniarum, right, which functions as the eye's photoreceptor, right? And it binds retinol, which is the light absorbing group, and it's shown right here where it binds retinol. Its binding site is really right within the lipid bilayer, right? It's shown in a magenta color. So each of the hydrophobic alpha helices is about 20 amino acids long, right? And you have alpha helices, and that's really what allows it to firmly anchor among the fatty acyl groups of the lipid bilayer, because it's highly hydrophobic. And then you'll have regions on either end that are slightly exposed to, one side of the membrane and the other side of the membrane, and notice they're more so of a random coil. Another structural motif that we see in transmembrane proteins is beta barrels, which are stabilized by inner chain hydrogen bonding. And you don't have the hydrogen bonding now with water that would happen in an aqueous environment, and instead they're inner chain within the polypeptide, so they're hydrogen bonded. One example is porins that allow certain polar solutes to really cross the membrane of gram-negative bacteria, such as E. coli, right? So what you have is one side of, actually let's just use the highlighter. You have one side of that porin that is basically hydrophobic because it interacts with the lipid bilayer. And then you have the inside, so it acts as a channel, and the inside of that channel that allows molecules to diffuse through is actually polar residues. So it has the central channel that is at least it has the central channel right here that's about 0.5, at least 7 angstrom wide, or 0.7 nanometers, and every second residue within this bilayer is typically hydrophobic, so it can interact with the bilayer. And the inside again could be polar. There's also aromatic side chains that are typically found in the lipid protein interphase, meaning they're found right here. Typically you have aromatic residues that can interact with the lipid head groups, right? And bacterial porins could be either monomers or trimers, right? They could be either monomers or trimers, right? And right here shown as a trimer, they're associated with one another through hydrophobic interactions. And this little white region is showing you the channel that allows molecules to diffuse through. So we have a channel right here that allows molecules to diffuse through. And it's the lining within the channel that dictates what type of molecules might diffuse through. The second type that we'll consider of membrane proteins are lipid-linked proteins, right? They're membrane-associated proteins that are covalently attached to lipids. They help anchor the protein really to the membrane. And if we think about lipid groups, or any other modifications such as glycoproteins, they typically can also mediate protein-protein interactions, or can even modify the structure and function of the protein that's attached to it. But in this example, we're looking at ones that help anchor the protein into the membrane. And they come in 3 different varieties or types. And a single protein can contain more than one linked type, right? Prenylated protein, fatty acylated protein, and Glycosylphosphatidylinositol, known as GPI-linked proteins. The first example that we'll look at is prenylated proteins, which are built from an isoprene unit, right? An isoprene unit is 5 carbons. You have a pi bond on either end of it, on carbon 1 and carbon 3, and you have another methyl. So they're built from an isoprene unit. The most common isoprenoid groups that are found linked to proteins are 15 carbons long farnesyl residues, or 20 carbon long geranyl residue, right? If you count the carbons, one is 15 carbons, one's 20 carbons. And the most common site for prenylation, where those groups are attached to a protein, and that is the site of attachment, R, right? Is a tetrapeptide that has the sequence CXXY, where C is a cysteine residue and X is typically an aliphatic amino acids residue, such as any aliphatic amino acids residue, alanine, leucine, isoleucine, and then Y. Well, Y is what influences the type of prenylation, right? Now, if a farnesyl residue is attached if Y is either an alanine, a methionine, or a serine, right? Whereas if Y is a leucine, then we get this 20 carbon geranyl residue, right? So proteins are farnesylated or generylated, really depending on the residue. And either one of those prenylated, either one of those isoprene, units, they're really added to the protein via an enzyme, right? So they're linked to the protein via an enzyme where, they're linked to the protein via an enzyme, and they're specifically linked to the cysteine residue. So what they form is a thioether linkage, right? So I'm gonna show you. Ta-da-da. So here's the cysteine residue, right? And that is the C terminus end of the cysteine residue. And then we'd have X, and then X, any aliphatic amino acids residue, and then Y. If Y was a leucine, then we'll have a geranylated, linked to the protein. If Y was either alanine, methionine, or serine, then it would be a farnesyl residue. And then once it's added, it's cleaved after that particular cysteine residue, it's cleaved at the C terminus end of the cysteine residue. So you have the cysteine. So suppose this is dash, and here's the cysteine residue. This bond is cleaved, right? And once it's cleaved, the C terminus end of the cysteine residue is methylated. And those prenylated proteins are really mainly anchored to either the intracellular membrane or to the cytoplasmic face of the membrane. Okay, I hope this was clear. Two types of fatty acids that could be covalently linked to membrane protein are myristic acid and palmitic acids, forming what we call fatty acetylated proteins. Myristic acid is saturated fatty acids, right? And it's biologically rare as it's 14 carbon saturated fatty acid. It's specifically linked to the protein via an amide linkage. Right? It's linked to the protein via an amide linkage to the alpha amino group of an N-terminal, typically a glycine residue, to the N-terminal amino residue, typically a glycine residue. Right? It's typically a glycine, right? And it's to the alpha amino group, the alpha carbon, and they're typically located in some, in subcellular compartment, such as like the interface of the membrane, the nucleus, the cytosol, the ER. Okay, well, the other type is palmitoylation. And in palmitoylation, what we have is a palmitic acid. It's 16 carbon saturated fatty acid, and it's linked via a thioester linkage once again. And then we have a cysteine residue. The other one that we talked about that involves a thioester linkage, this was a thioether linkage, right? We have a thioether linkage. In this case, we have a thioester linkage to a specific cysteine residue. Well, it's thioester because palmitic acid will have the carboxylate, right? Anyway, so you'll have the carbonyl oxygen and then the sulfur from cysteine, so it's a thioester, right? Meaning, okay, well, let's just not draw it. All right. Well, palmitoylated proteins occur almost exclusively on the cytoplasmic face of the plasma membrane, and they typically participate in signaling. Unlike myristic acid, palmitated proteins or groups to a given protein can actually be removed by the action of an enzyme known as palmitoyl thioesterase. And those proteins, um, well, anyways, by an enzyme known as palmitoyl thioesterase, which may suggest that there's this reversible palmitylation, meaning that a palmitoyl group might be added and then it can be removed, might help regulate the transmembrane signaling, because it can regulate the association of the protein with the membrane when it's linked to a palmitic acid. And then there's the enzyme that cleaves that bond. So it can help modulate the overall signaling process, right? Palmitoylated proteins participate in signaling, right? And because palmitoyl thioesterase can cleave that bond between the fatty acid and the protein, which in turn, it can really regulate the association of the protein with the membrane if it cleaves that bond. And it's believed to help modulate the signaling process. Okay, well, another type that we'll take a look at is glycosylphosphatidylinositol, or GPI-linked proteins. They're really located only on the exterior face of the plasma membrane, much like glycoproteins and glycolipids. Well, if we take a look at its overall structure, the core structure consists of a phosphatidylinositol, right? So, notice that's would be an example of the glycerol. You have a fatty acid, you have a fatty acid, you have a phosphate, so you have a phospholipid, a glycerophospholipid, and in this case it's attached to an inositol group, right? Well, that is basically the core structure of it, a phosphatidylinositol. And then that phosphatidylinositol is linked via a glycosidic bond, right? It's linked via a glycosidic bond to a tetrasaccharide unit, right? And in this case, it's composed of 3 mannose residues and 1 glucosamyl residue, where you have a glucose that has an amine, right? And then the mannose that is found at the non-reducing end, right, is forming a phosphodiester bond, right, with basically a phosphoethanolamine, right? And this phosphoethanolamine in turn is forming an amide bond with the protein. And it's specifically linking the protein at the C-terminus end of the protein. Right, so it has several different components. C-terminus. Okay, I'll spell that out. So now, the identity of the protein can dictate the identity of the core tetrasaccharide. So really, depending on what the protein might be, the core tetrasaccharide unit might vary or can vary with the identity of the protein. And if you consider the 2 fatty acids in the glycerol phospholipid, they can have considerable diversity as well. Okay. Our last type is really the simplest, peripheral proteins, which are known to associate via electrostatic interactions and hydrogen bonding with either hydrophilic domain of nearby integral proteins or with the head groups of the membrane lipids. And in the lab, if you wanted to release peripheral proteins from their associated membrane, this could be done by really relatively mild treatment of um, that really what it will do is interfere with electrostatic interactions and hydrogen bonds, right? High salt concentration or changes in pH. A common reagent that is used is really carbonate at a high pH. So this concludes our discussion of peripheral proteins, lipid-anchored proteins, and integral membrane proteins.